So it's a pleasure and privilege to talk to you today. So this, again, is an educational part. And also, it's a very broad title, so I'll need to restrict the topic to something which is manageable within the time that we have. And so here's what I'm going to be talking about. So I'll give some examples of sensing technologies that hopefully can be combined, already are being combined with MRI. And specifically, I'm going to be talking about calcium and voltage imaging. But just remember that neuronal activity is a kind of very broad term, so there are many different aspects to neuronal activity. And this also includes things like intracellular signaling and oxygen, metabolites, and many other things, and gene expression. So all these things can be sensed, and due to Brain Initiative, we have lots and lots of tools. So what I'm going to be talking about is completely non-exhaustive list of things. And then I'll also talk a little bit about manipulation of neuronal activity of cells and networks. And this can be electrical, optical, chemogenetics. I'll talk about combined opto and chemogenetics, something that you may not have heard yet. But again, there are other ways of doing this. There's ultrasound and many other things. So before we get to these topics, let's just take a very quick look in terms of what's inside neuron. And this is going to be helpful because this is how we're going to realize what actually is getting modulated. If I'm being too basic, my apologies. This is an educational topic, and I realize that for some of you, this is going to be way too trivial. So I'll mostly talk about the machinery that is available in neurons, but there are obviously some other cells in the brain, such as astrocytes and vascular cells, and this is important for those cells as well, at least part of this machinery. So we'll start with the ion channels that control membrane excitability, and the question really is, what is it that makes neuron excitable? Why neurons are excitable? So they're electrically excitable because they have this array of channels. And so here are different channels for different ions. And the ions, the kind of majority of ions that we need to take into account is calcium, sodium, potassium, and chloride. And these channels are passing these ions with high degree of selectivity. And they can be operated by different things. So one of them is voltage. So for example, voltage-gated calcium channels are very important for calcium transients that are associated with spikes. Voltage goes up and there's a calcium increase. But there are also some other mechanisms. For example, there are calcium-dependent potassium channels. And this is sort of like a brake. When you have high concentration of calcium, neuron is very active. It opens potassium conductance and there is a hyperpolarization. Now, when we talk about the membrane potential, so this is a famous Goldman-Hodgkin-Katz equation so this equation tells us how to calculate the membrane potential. And so the membrane potential is going to be dependent on some kind of constant. And this expression in here, where P's are partial permeability, relative permeability of different ions, and this are concentration of, for each ion, there is a concentration of this ion outside and inside, so always outside and ion inside. So now you can see from this expression that when one of ions have more permeability than others, then this particular term, let's say for potassium, is going to be dominant, and then this is what's going to determine the membrane potential. So for each one of the ions, there's this Nernst equation, and this tells us what's the equilibrium potential for this particular ion. And here what they are under normal conditions. For sodium, it's positive. It's about plus 60. And so I need to mention that the resting potential is about minus 70. So that means that if we open conductance for sodium, we're going to get depolarization, right? If we open conductance for potassium, we're going to get hyperpolarization. So this is how it works. And so now with this in mind, channel rhodopsin is just another ion. It's another ion channel. So it's an ion channel mostly for sodium. And so looking in here, because sodium is the equilibrium potential is positive, much more positive than the resting potential. This is why we get depolarization. So now what else is out there? Well, so we also have ionotropic receptors. So ionotropic receptors are also ion channels. And here are some of them. They're also ion channels, but they're operated by ligands, for example, glutamate. So when glutamate binds to an NMDA receptor, we get currents. When the glutamate is not present, the channel is closed. And there are other channels as well, so there are a few of them indicated here, but there's obviously more. So now, in addition to this, there are also metabotropic receptors for neurotransmitters. And these are known as neuromodulators. And so why are they neuromodulators? Well, this is because their time course of their action is slow. So it's much slower than what we're used to, in terms of channels and ionotropic receptors. So channel is fast. Remember that channels, for example, sodium and potassium channels, is what makes an action potential. And the duration of action potential usually is just 1 millisecond. So in here, we talk about neuromodulation. All those guys that work through G proteins, which are part of intracellular signaling machinery. And they couple to other intracellular bits and pieces through different pathways. So for example, here we can couple to increase of calcium through release from intracellular stores. And others, depending on the type of the G protein, couple to other machinery that have intracellular kinases and phosphatases. And here's the key. And the key is that this kinases and phosphatases, they feed back, so they regulate the fast neurotransmission. And so now, because we also want to be scholarly, we need to mention Paul Greengard. And he got his Nobel Prize in year 2000 for discovery of this type of network that integrates fast and slow neurotransmission. So now, with this in mind, let's take a look of how we can sense calcium and voltage. So now I'll start with calcium, and this is because calcium sensing is where, for many years, but especially recently, due to the Brain Initiative, we have amazing advances. And there are many names to name, but this is one of recent papers, which is kind of like a shock and awe paper in terms of the field of view and the signal to noise and the spatial resolution and number of cells that we can cover. So this literally gives you an access to 1 million of neurons in the live mouse cortex. This is a view from the top, so this is the entire mouse hemisphere from top. And the way that this is done is that they came up with a little glass dome. So what they do is that they take half of the skull out and they replace it with a glass dome. And through this glass dome, they can now look at all these neurons all over the hemisphere. Now, it's optical imaging. So remember, MR is very good in looking, at any depth, right? But optical imaging is limited to the penetration, so we cannot see very deep. However, we can see lots and lots of cortical neurons. And here's an example of segmentation of this bunch of neurons within this region, and these are their calcium time courses. And each one is related to spikes. So there's a way to extract spiking information from these calcium transients. And here is a movie which is pretty amazing. So, when I look at this, I'm really shocked to see this, to tell you the truth. But this also may be something that underlies our resting state MRI signals. So this is why this is, has particular importance for functional MRI. So now we can also image deeper using the same calcium sensors, but the problem is that the penetration is limited. So the way that it's normally done is that we need to remove tissue on top of this. So in here's a very impressive paper from Xue Han, and she's at Boston University. And they're looking at hippocampus through a cannula. So they actually, they take out a piece of cortical tissue and they install a cannula, and now they can look through. And so here, what they can do is that they look at the surface of the hippocampus. There are lots and lots of cells. And also, it happens to be that these neurons, they don't fire at the same time. So there is a spatiotemporal separability, and this can be used to extract this region of interest, which are individual neurons. So this is how they also can get hundreds of neurons, in this case in hippocampus, and these are the time courses. And here is a movie. It's a movie where a mouse is doing a conditioning task. So there's a stimulus, there's a delay, and there's a puff of air, I think, on the mouse's eye. But there's also lots of spontaneous activity. And by the way, you can also see blood flow in here. So you can see that there's some erythrocytes going around. So now, I already mentioned the resting state. So here is a very interesting paper from Elizabeth Hillman from Columbia University. And I'll tell you a little bit of what they did in here. So what they did is that they found a way to do imaging of calcium using a camera looking at the top. So again, both hemispheres are available. And also, at the same time, they look at the total hemoglobin. And so now what they do is that they ask a question, well, can we find an impulse response function that would look conventionally, what we expect from functional MRI studies, and use it for convolution, and then try to predict the signal and see how it compares to the actual one and look at the residuals, right? And so I think that you probably agree with me that the fitting is very, very good. So there are some residuals in this. I need to mention that in this particular mouse, the calcium indicator was only expressed in pyramidal cells in layer 5. So it's one single type of pyramidal cells. So this signal would not have any of the neuromodulation in it. And the residuals kind of have this broad spatial pattern that look like neuromodulatory signal. And I want to mention something else. So, as a neuroscientist, when I look at this movie, I think it's amazing. And so what's amazing about this is that if you go into neuroscience textbooks and you look for what do we know about this type of neuronal activity that is spontaneous and that's mostly symmetric in both hemispheres, you're going to find nothing. So there's really nothing known in terms of basic neuroscience why this type of activity is there. And before this movies became available, if you would find, big neuroscience figures at the Society for Neuroscience meeting and ask them, do they think that this type of activity exists? The answer would be no. And this is because we know that, our resources are limited, and so we don't have any energy to waste. So the idea of that being ongoing oscillations that would be symmetric and keep working around in the cortex is extremely strange from the basic neuroscience perspective. However, this is where the technology comes and shows this perspective. So this is something we didn't know before. And now this is extremely important on its own and also extremely important for the resting state functional MRI. So I would make a point that if we now want to go and understand more about the neuronal correlates of those functional imaging signals that we call resting state, we really need to go to the basic neuroscience of this phenomenon and try to understand what that is. So voltage. So obviously we can measure voltage using classical electrophysiology, and this has been done, and even combined with functional MRI. So here's Nikos Lagathidis. He did a pioneering work on this. And here is an example from one of their papers by Amir Shmuel, who's the first author. And here they were looking at the positive and negative bold, and there was an electrode inserted in a certain place. And now by looking at different patterns of visual stimulus, they could manipulate where this activity is on the cortex, such that sometimes the electrode would be in the positive region and sometimes in the negative region. And so this is how they came up with the conclusion that there's a decrease in spiking in the region of negative bold. So this, of course, is very impressive, and it's one of the classical studies. However, there are a few limitations. So one of them is that some filtering is necessary, and this is because there is a crosstalk, there are artifacts due to gradient switching. And the other problem is that there's no specificity, so to cell type. So we can talk about overall spiking activity, but it's difficult to talk about the circuit. And this is why, obviously, in neuroscience, people knew this limitation for many, many, many years, and people have been working on optical ways to sense voltage. And this started with this demonstration in the late 70s of optically recorded action potential in the squid giant axon. Here are some people that were pioneering this field, and here are the same guys a few years later. And this is the first in vivo image, optical image using voltage sensitive dyes from late 80s of visual orientation columns in a cat. So now voltage-sensitive dyes are hard. And the reason that they're hard is because voltage is very rapid, right? So I just said that the duration of action potential is 1 millisecond, so that's very fast. And also the sensor needs to be physically in the membrane, and so there's only so many dye molecules that we can stuff into the membrane without changing membrane properties. And this is one of the reasons why the voltage imaging technologically is lagging behind calcium imaging. But nevertheless, there are, again, mostly due to right now Brain Initiative, there are good advances in voltage imaging, and especially in the genetically engineered voltage sensors. So this, of course, is not something for you right now to read, but I'm just putting it there so that you have this reference, and if you want to, you can go back and take a look. What type of sensors are out there? And I'll show one example. So this is again from Mark Schnitzer lab, although I have to mention that there are many, many good other names of people that work on the voltage sensing. And here what they do is that they did sparse encoding of voltage sensor. And here is one neuron that is responding to the task, and the task is a visual task. This is optical imaging using a CCD camera, and there's scattering in the tissue, so the image is a bit fuzzy. And also the field of view is small because they need to image super fast. And so now you can see that when they look at the spiking of this neuron, so now this is optical recording, you can see that there is still direction preference, so it's not responding here, but it's very responsive here. And this is the same as a little movie, and again, it's not as kind of gorgeous as calcium imaging because we need to image so fast, and so we cannot afford very large region of interests. And so now, a few words about the manipulation of neuronal activity. So I'm not going to talk about electrical, but I invite you to take a look. This is from Saturday. So last Saturday, Ian Shih gave a very good talk, and he was talking about deep brain stimulation and also about his own results. So this is a very good resource. But I'll say a couple of words about others. So optogenetic is, again, something that probably all of you know, and most likely I don't need to introduce it too much. But these are figures from, few of the, by now, classical papers. So this is the first generation of opsins. This is channel rhodopsin that comes from this algae. And this is another one of the same kind that comes from this little algae colony. And this is a proton pump, halorhodopsin, which comes from halobacteria. So now all of these channels and pumps, they are exogenous for mammals. We don't express them naturally. And so this is why it's, we need to use transgenic technology to insert this into our genome. And so here is an example of a mouse that expresses one of those in cortical cells, I think, or maybe an olfactory bulb. And you can see that for each one of the light pulses in here, we can get a spike. And so this, again, these are a few of the first papers. And now there are lots of different variants, both excitatory and inhibitory. And there's a lot of spectral choice, and there's also a lot of choice in kinetics. Chemogenetic stimulation. So chemogenetic stimulation has been there for a while, but the real breakthrough was made by Brian Roth that generated these dreads. And here is a very good paper. It's a review paper. It gives also good introduction and good understanding of limitations. It's called Dreads for Neuroscientists. And so dreads are G-protein coupled receptors. So if now you remember what we just said about G-protein coupled receptors, these are slow, and they also couple through the intracellular machinery. So the G-proteins, they regulate neuronal excitability by this interaction between the fast and slow neurotransmission. And the way that they're made is that they have, these are the G-protein coupled receptor that has part outside of the cell and part inside of the cell. So the part outside of the cell is a door, and the transmitter comes and does knock-knock. And so now on the other side of the door, there is a G-protein that says, oh, somebody's knocking on. And so now the G-protein is going to go and do its G-protein-like things. And so now what these dreads are is that they don't recognize, they're genetically modified such that they don't recognize any of the endogenous neurotransmitters. So there's no crosstalk. However, they're made sensitive to something, to small molecules that are exogenous and normally are not present in the system. And now their intracellular part is exactly the same as those normal, naturally occurring G-protein coupled receptors that couple to G-protein of different kinds. And some of them happen to be overall excitatory, and some of them happen to be overall inhibitory. So this is a summary about G-proteins. So they're engineered G-protein coupled receptors. They're activated by inert small molecules. That means that they don't have any other action except this. So now this is important. So they're non-invasive in a sense that for optogenetics, we need to deliver light, right? So we need to, in most cases, stick into the brain on optical fibers. So here we don't need to stick anything in the brain. We can inject this compound IP or IV, and it's going to work, but it's slow. So it's slow. So it's going to take about 20 minutes before it starts working, and then it's going to work for about 2 hours. The spatial coverage is determined by the pattern of expression. So this also is different from optogenetics, where the pattern is going to be determined by where you can get light. And also the cell specificity is available due to genetic targeting. It uses this G-protein signaling, this intracellular signaling that sometimes is difficult to control. And also transgenic dread mice are available. And this is a break. And again, I refer you to this Ian Shee's presentation on Saturday, where he also gave a very good introduction and shared some of his own work on the chemogenetic functional MRI. And so now I want to, in the last few minutes, I want to talk about something that you maybe not heard about yet. And so this it's called luminopsins. And so the idea is that we do need to get a fiber. Optogenetics is good, right, because it's fast and it doesn't rely on the intracellular G-protein coupled machinery, which is difficult to control. However, we need to deliver light. So how do we deliver light? Well, we stick a fiber in, but what if we want to activate 5 different areas? So are we going to stick 5 different fibers? Well, that's very hard to do and very invasive, right? So why don't we genetically encode light, right? So can we do this? Well, nature can do this. So I'm sure that you all are familiar with this picture. So this is a blue tide, and so those blue tides are caused by this little marine microorganism that has an enzyme that's called luciferase. And luciferase produces light when it has substrate present. So this is not the only organism that does it. So here's another couple of examples from nature. So now can we harvest this and can we harness this for our purpose? And the answer is yes. So, so here's Ute Hoogschwander. Sorry if I mispronounce her name. And she's the mastermind behind this luminopsins. And so what she did is that she integrated the optogenetics and chemogenetics by making these chimeric proteins that have both the optogenetic actuator and the light emitter, the bioluminescent light emitter. So before, to start with, can we look at bioluminescent proteins in a mouse? And the answer is yes. So of course you can do this. So here is a mouse that expresses a bioluminescent protein. And now if the substrate is delivered, so the blue line is IV, so that we can see that the light is emitting for about 15 minutes. If we do it IP, so then the intensity is lower, but the time course is longer, so it depends what you need. And here's a proof of principle demonstration from Ute's lab that this actually works in neurons. So here are 2 types of neurons. This is a neuron that expresses, it expresses the luminopsin of excitatory kind. So it's coupled to excitatory opsin, and this one is coupled to inhibitory opsin. And now when you deliver substrate, so this was done in brain slices or possibly in cultures, You can take a look at this paper. So when you deliver the substrate, in this case, there's an increase in spikes, and in this case, there's a depression decrease in spikes. So here are a few things about luminopsins. So this is a fusion protein of luciferase and opsin, and they're activated by luminescent, but also they can be activated by external light. So there is more flexibility in here because you don't lose the property of the natural opsins, but you can also activate bioluminescence. You can silence or you can activate neurons, so there's bidirectional control. There is cell specificity because you can do genetic targeting. It doesn't rely on intracellular signaling. It supports multiplexing because there are luminescence, you can genetically engineer it to be of different colors, and luciferases also come at different colors, different types, and they have different substrates. And so future, and this is something that Uda is currently working on, I believe, you can actually take the luciferase and opsin and separate between the pre- and postsynaptic terminal. And so now you actually have a synapse that you have total control over. And this is just in the last few seconds, I want to share with you a few slides, two of them, that Uta sent me yesterday. And so here's a demonstration that this type of thing works in vivo. And here's a mouse. They inject a substrate. They inject it in substantia nigra, in basal nuclei, in a place that is known to induce certain type of behavior. And the behavior is that the mouse starts circling. And so here's when they inject vehicle, and the mouse is just doing normal behavior. And so here what happens when they inject the substrate. And the inhibition or excitation causes rotation in different directions. So that's also kind of funny. And so now this is their rendering trajectories of the mouse from their experiment, and this is a baseline. And here is intravenous injection of substrate. So you get the idea. And so lastly, I want to say that, I didn't cover anything of our own work. However, on Saturday, I also gave another talk, and so in there I was talking about work from our own lab. So you're welcome to go and take a look if that's what you're interested in. And thank you very much.